Hi, and welcome to your virtual Microscope Lab. This is an activity where you get to learn about the microscope in a little more detail, some features about how a microscope works, and also a little insight about how to prepare things for viewing under the microscope. So as you follow along with this video, I encourage you to write things down. There is a lab handout. So you probably want to have a copy of this lab handout so that you can make notes of important things as we go through the lab. And also, since this is a video, you are encouraged to pause as needed so that you can write things down and really understand the concept that we are discussing. So I will be showing you a combination of prep view video clips as well as what it looks like through the view of the microscope. So I hope you enjoy this introduction to microscopes. So the lab that you're doing today is actually going to be done with a slightly different type of microscope than what we learned in class. This is still a light microscope, but notice it has two eyepieces, so it's called a binocular microscope. And the stage clips are a little bit different. You will notice this when I put a slide on when I'm demonstrating. The slide is actually going to be placed in the clip like this instead of under the clip. And then the other feature that's different is that there are these wheels to turn, and notice when I'm turning the wheel, it's moving that slide from side to side, and then if I turn the upper knob, it is going to move it front to back. So this is how I will be manipulating the slide on the microscope during your lab today. So the first slide that we're going to look at under this microscope is just a simple letter E. So this is a letter E that I have printed on just plain white printer paper. I hole punched it out and then it is taped to a slide. So when I put it on the stage of the microscope, I'm going to have it facing me the way that I would normally read it. And that position is really important for the lab that we're going to do today. So when I put this on the slide, again, it is oriented so that if I were looking down at it, it would be right side up and facing the correct way. And here you see what that letter E slide looks like under the microscope. So this is on the 4x objective lens. If we go up to the 10x objective, which is medium power, we get a more zoomed in view. Now notice I need to adjust the focus here. I'm using the fine focus knob now that I'm not on low power anymore. And I'm just going to center that E a little bit and maybe increase the amount of light to make it a little bit easier to see around the edges. So when you are drawing what your letter E looks like under the microscope under 10x, this is your view. What you're looking at here is a side-by-side view of moving the slide away from you and then reversed and moving back towards you. In this comparison, the microscope slide is being moved to the right and then being moved to the left. So we're going to make a wet mount today with a piece of absorbent cotton. So the way that you want to do this is take your microscope slide, and we're going to lay that on the lab bench. And since it's called a wet mount, that should clue you in we're going to need some water. So I just have a beaker of plain tap water here. You could use distilled water if you're concerned about impurities. And go ahead and put a drop, not too much, a little bit goes a long way. Put that on your cover or your microscope slide so you can see about the amount of water that I put there. And then whatever you are making your wet mount of, you want to put in that drop of water. So I'm going to take my cotton ball and I'm just going to tease off a few threads. Remember, we're putting this under the microscope, so it is going to be really magnified, so we don't need to put that whole cotton ball on there. Now we have what's called a cover slip. A cover slip is a little square of glass. They are glass, so they will break. And this square, you are going to lower gently into your drop of water. And I normally try to lower it from one side to the other so that we don't get a bunch of bubbles trapped in there. So now you can see our cover slip is on top. The water has spread out. We don't really have any bubbles in there. If you want to tap on it with your finger just to get it all nice and flat, you can certainly do that. But there is our cover slip on top of our wet mount. So here are the cotton threads under the microscope. This is the view that you would see when looking through the eyepiece. And these cotton threads are currently being viewed under the scanning power, which is the same as the low power or 4x objective lens. So if we go up to the next highest power and go up to that 10x lens, we're going to have to adjust our focus. Again, making sure that we are using the fine focus knob here because we don't want to crack the lens or the slide that we're using. And so notice that it's kind of difficult to get these threads into focus. There's a lot of really bright white in the background because of the amount of light coming through. So if we increase the amount of light, notice how that gets so much more difficult to see. So this is important when you're using a microscope. If you're trying to look through something that's swimming in just clear water and there's a lot of light getting through, that is probably going to inhibit your view of what you're trying to see under the microscope. Now for this section of the lab, instead of using sheer ribbon, I just have 3 different threads of that ribbon. Because when I tried to make a wet mount with the ribbons, it was too thick and it was not very easy to see. So this is a modification from your lab sheet, but it should still work the same way. So this is what those 3 colored threads look like under low power. There are 3 colors here. There is a very light green, almost a yellow color. There's a blue and there is a pink. So if we go from 4x, which is what you're looking here, up to the medium power 10x objective, we need to adjust our focus, and you can see we're much more zoomed in on these 3 threads right now. So hopefully you can tell the difference in color, that yellowish green, the blue, and the pink. Notice that even on medium power, as I bring this into focus, it's really hard to tell which one is on top of the other. And so if we go up to the highest power, we still need to fine tune our focus, but look how zoomed in on these ribbons or the threads that we are now. It is very difficult to bring any one of them into focus while the other ones are also in focus. So as I am adjusting the focus, it looks to me like I can bring this one into focus that's on the bottom, which is the green, and then the blue I can slowly bring into focus, and then the pink one. So notice that all 3 of them cannot be in focus at the same time. This is kind of difficult for you guys to appreciate through watching a video, since you don't get to actually manipulate the focus of the stage and see the stage going up and down. However, hopefully you can at least see that when the blue thread, which is the clearest color to see, when that is in focus, the other ones are not. And so you have to focus down through different layers to try to get the other threads in focus. So this is demonstrating to you the depth of the field of view, and this is really important if you're trying to find something under the microscope that's swimming. It might be swimming up vertically in your drop of water, even though it seems like your drop is really flat. And so that's when you are really going to see the impact of the depth of your field of view. This is a common method for staining a slide when you already have a wet mount prepared. I have some iodine here, which is a starch stain. It will stain starch a dark purple. And so as you can see right now on our slide, there's starch in there, but you don't see any purple color. So this would be very hard to see under the microscope. So we're just going to remove the dropper from the iodine and just place a drop here on the edge of the slide. Now notice that it starts to soak through, and in order to help that iodine get pulled through, if we take a piece of paper towel on the other end and put it right on the edge of the cover slip, it should start to soak up some of that water from underneath the slide. You can see it starting to soak up here. And as it soaks the water up from one side, it is going to pull that iodine through to the other side. So now you can see that iodine has moved through the slide, and it actually, you can start to see some of that color. So you can see some of the dark color there of the iodine coming into contact with the starch. So that is how we know we were successful. So here are the starch grains under low power. This is under the scanning or the 4X objective lens. And as you can see, there are a lot of starch grains here. I definitely do not expect you to be able to count all of these, so don't worry about that, but you can just describe the number of starch grains as too many to count, or that there are very many starch grains. Hopefully you can tell the color here, kind of a purplish color, a little bit black around the edges. That just has to do with how the light is passing through the specimen. And then we're going to go ahead and switch up to the medium power. So this is the 10X objective. We need to fine tune our focus using that fine focus knob. And here you can see there's a little bit better view of those starch grains. You can see their color a little bit better, especially the ones that are well lit in the center. And you can see a little more definition. Now this is just medium power, so again, if we change our objectives and we go up to high power, now we definitely need to adjust our focus, and notice how dark it is. So I'm actually going to increase the amount of light a little bit. See how that's lightening it up, and that's going to allow me to better focus on these starch grains. So notice that there are a lot fewer visible here. Also notice, just like we talked about in the colored threads, it's difficult to focus on all of them at the same time because of that depth of the field of view. So this is going to be what your starch grains look like on a high power or 40X objective lens. So you can use this as your high powered sketch. This should be a little bit more countable in terms of the number of grains than it was under low power. So we're not looking for an exact number here, but just give it your best guess as to how many starch grains are visible under high power. For this last section of the microscope lab, we just have a colored photo. This is cut from a magazine, so it's on magazine paper, but you can see it's just a colored image. So this is what it looks like not under the microscope. And we are going to now place it on the microscope under the 4X lens, and let's see what it looks like. And here is our colored photo under that 4X objective lens. So you can already tell a major difference between what it looks like magnified versus what it looked like before we put it under the microscope. So we can definitely see how the colors are combined together, even on this 4x objective. But if we move up to the next higher power, which would be our 10x objective, we need to adjust our focus. We can see how that color is deposited even clearer. And then from here, we can even go up to the 40x objective and focus in, and we can really see those individual colors. So if I move the slide around, we can look at different sections. This looks really cool. It's almost like tie-dye when you look at it under the microscope versus when you just looked at that color photo. So this gives us a little bit of an insight as to how those colored photos are printed, so you can see it on the microscopic level how the colors are combined. So this method of making a dry mount is not your typical way of making a dry mount. In fact, we hardly ever make dry mounts in biology class, but I did want to demonstrate it for you because it was explained on your lab sheet. Now the reason why we're making our dry mount this way is because our specimen is this feather. It's from a feather boa, so it is bright red instead of a natural bird color. But it is actual feather, and it's so thick that we can't really make a wet mount out of this and hold it down with a cover slip. So this is a very simple method of simply putting the feather on one slide and then pick up a second microscope slide and put it on top. Now notice when I do that, it's holding down the feather and weighing it down so that it's going to hold it in place when I put it on the microscope. So here's our view of the feather under the 4x objective. So it's really neat to see these delicate structures of feathers and how they're able to catch the air when the bird is flying. So our dry mount here, because of the way we prepared it, we can go up to medium power, which I will do, and we can fine tune the focus here. But this is as high of a magnification as we can go, because if we try to go up to that 40x objective, the length of the lens is so long that it would hit our dry mount slide, since we had to use 2 slides thickness instead of a normal cover slip. So this is your 10x view of that feather. So go ahead and sketch this one for your microscope lab. So I hope you enjoyed watching these video clips of how to use a microscope. And if you have any questions, please feel free to reach out to me by email or attend the virtual office hours for extra help.